Okay, so we're very excited today to have Dr. Mallory Brayer. She's a newly minted PhD from George Washington University, and the title of her talk is The Intertwining Roles of Mechanosensitive Channels, Cellular Stress, and the Proteasome in Beta Cell Function. And a little bit about her. She received her PhD in biological sciences from GW University, where she specialized in cell and molecular biology. Her research focuses on the interplay between proteasome function, mechanosensitive channels, and cellular stress in pancreatic beta cells, particularly as it relates to type 2 diabetes pathogenesis. She has anchored multiple peer-reviewed publications on beta cell proteostasis, amylin segregation, and mechanobiology, and has presented her work at leading conferences, including the Gordon Research Conference and Keystone Symposia. As a scientist living with type 1 diabetes, she brings a deeply informed and personal perspective to her investigation of beta cell biology. Thank you so much, Mallory, for joining us. We're very interested in this work, and it kind of, follows after some of the talks we've hosted, particularly with the channel functionality, how it works and how it works in normal beta cells and those who are undergoing the progression to type 1 diabetes. So this is going to be a little bit of a type 2 flavor, but still kind of
Yep.
digging into the mechanism, which we always are very
Exactly. I mean,
interested
most of my work is very much beta cell focused, so hopefully a lot of the mechanisms we could still see the interplay there. So
in.
that's
I think
always the goal.
that's the goal. And people can drop questions in the chat as the talk progresses. Thanks so much, and take it away.
Great. Thank you so much for the introduction. I do appreciate it. You've already heard the title of my talk, and as you could probably see, there's a few different things going on there because I've worked on quite a few different projects throughout my degree. So just to kind of give you an outline of where we're headed, because there's so many different moving parts here, I'm going to give a little bit of a background on just pancreatic beta cells in general, and as well as in the context of type 2 diabetes, then move on to kind of the first project, looking at the kind of interplay between hyperglycemia and pro-inflammatory stress, and what changes these causes in pancreatic beta cells, really looking at the transcriptional changes, then moving into this regulation of proteasome activity and how it influences beta cell identity, as well as glucose sensing, and having this, I mean, beta cells are the ones that sense changes in blood glucose in order to secrete insulin. So if we have changes to that process, we have changes to their functionality. And finally, we're going to move into this last project that's a little more collaborative with a biomedical engineering group and looking at mechanosensitive channels, so getting a little bit into mechanobiology and seeing what effects that therapeutic ultrasound can have on pancreatic beta cells in the context of potential treatments for type 2 diabetes. So, as I've said to begin with, a little bit of a background. Well, the pancreas itself is a retroperitoneal organ. It has a lot of different functions, largely because it has two different tissue types. We have the exocrine tissue that makes up the bulk of this organ. This is what's producing different digestive enzymes and helping break down things like proteins that enter the intestinal tract. The endocrine tissue is scattered throughout the whole pancreas in these islets of Langerhans, where they make these little kind of islands throughout the pancreas. These are comprised of a whole different mess of cells. The ones that make up really the majority of these islets are the alpha cells, beta cells, and a little more minorly, the delta cells. All of these have different roles in helping regulate blood glucose. That's the primary job of these endocrine islets. What I've been really interested in are the beta cells, the ones that are producing these two different peptide hormones, the first being insulin, which is the one most people are pretty familiar with. It's the one that is secreted in response to rises in blood glucose. Specifically, when blood glucose rises, we have these cells bring in that glucose, metabolize it into ATP, and this ATP is then used for this calcium-mediated insulin granule exocytosis. So we need to have this really glycolytic process in order to have insulin secretion. Once this insulin is released, it goes to peripheral tissues, things like skeletal muscle or adipose, where it'll help these tissues actually bring in that excess blood glucose. That way we don't have this excess sugar in circulation and causing vascular damage. And it also lets these peripheral tissues use that energy that's available to them, whether it gets stored as something like glycogen or gets used immediately and converted into something like ATP for immediate energy. The other pancreatic beta cell peptide that gets talked about a little bit less is this amylin, or this islet amyloid polypeptide. You'll see it abbreviated by IAPP a lot throughout this talk. This is something that doesn't play as big of a role directly in blood glucose regulation. It indirectly does in the sense that within these islets, it'll help modulate the activity of these other cells, whether it be alpha cells or beta cells, and help regulate insulin and glucagon secretion from these cells, respectively. It also has quite a few receptors within the hindbrain and actually plays a role in appetite regulation as well when it's viewed systemically. In the case of type 2 diabetes, well, let's take a step back. Diabetes itself is a pretty heterogeneous disease. They all share the common characteristic of some type of chronic hyperglycemia. The exact causes differ quite a bit depending on which disease you're specifically looking at. In type 1, we think about things like autoimmunity and this kind of nearly complete destruction of beta cells. While in the case of type 2 diabetes, we're looking at beta cell populations still being there, but they're not able to produce enough insulin to really meet the metabolic needs of this individual. And this can happen through quite a few different processes. There's genetic components, there's lifestyle components, there's environmental components that all contribute to this loss of blood glucose regulation, leading to this dysglycemia and more significantly, hyperglycemia. At a systemic level, we could think about peripheral insulin resistance. So these target tissues for insulin aren't able to respond to insulin anymore. Generally, it's through a decreased expression of these insulin receptors on their plasma membranes. They're not able to take up this insulin, so they're not being able to take up this excess blood glucose, so it's staying in circulation. And this happens even when there is still enough insulin being produced, or even too much in the case of something like hyperinsulinemia. We have a lot of insulin, but the peripheral tissue's not able to respond to it. At the beta cell level itself is where a lot of my work comes in. In the case of the beta cells themselves, though, we're looking at quite a few different processes that contribute to their failure. First and foremost, in really the earliest stages of disease, we see these cells become what's called glucose blind. They start reducing their expression of these glucose transporters along their plasma membrane. They don't have glucose receptors, so they don't know there's changes in blood glucose. As far as they know, we're at a constant euglycemic level, even if there is more blood sugar in circulation. So that's one way that we have this loss of functionality. And with that, if we don't have them sensing there's glucose and they're not taking up that glucose, they're losing the ability to produce ATP and subsequently have this calcium-mediated insulin exocytosis. And we can have impaired insulin secretion through a couple other steps throughout this process, whether we're looking at membrane depolarization or processing of insulin granules. They're not able to produce as much insulin effectively. And as this process continues to occur, these cells do become stressed. They can have predominantly reactive oxygen species stress, really contributes to their ultimate demise as cells. So in order to try to minimize that stress, they can actually start downregulating certain transcription factors and take on a phenotype that's a lot more senescent. They look a lot less like beta cells and aren't really producing as much insulin. And therefore, they kind of go through this almost de-differentiation. And finally, we could be looking at cell death, where this stress contributes to really apoptosis and pyroptosis that ultimately reduces the functional beta cell mass and therefore the capacity of that individual to produce insulin, even if they were having, let's say, a perfect lifestyle, perfect environmental factors. They've lost so many beta cells, there's not enough of them left to really keep up with the needs they have. So to start kind of earlier in the disease states here, we're going to look at some pro-inflammatory signaling and see, well, how does this really contribute to this beta cell failure? There's been a couple different papers out over the past probably 25-ish years that have different viewpoints on this that seem to vary a lot based on what cytokines you treat with, what glucose concentrations you treat with, and what model organism you're using. So this may be slightly different than some of the other results that have been previously published in other model systems. But what we generally see is that during the progression of type 2 diabetes, we start to have innate immune cells really start affecting the islets. Within the pancreatic islets themselves, there's usually resident macrophages that just kind of stay there and will help kind of, if there's any issues with the cells, if something needs to be degraded or turned over, they'll help clear out damaged tissues. However, we start seeing in type 2 diabetes, these macrophages take on a more pro-inflammatory phenotype. and begin recruiting additional innate immune cells, largely through the release of these danger-associated molecular patterns from islets. These can be things like fragmented DNA or protein aggregates or misfolded proteins that overall signal to these immune cells that there's something wrong with these cells, we have to do something about them. What generally happens is we start seeing inflammation within these islets, and this promotes interleukin 1 beta secretion from the islets themselves as well, especially from the beta cells. And as these cells take on this pro-inflammatory phenotype, start producing cytokines, we also see a reduction in insulin secretion from the beta cells on an acute phase. And less acutely, when we look at more of a chronic disease, this inflammation ultimately results in apoptosis and pyroptosis, really just reducing functional beta cell mass. So we want to ask here, how do these different kind of stressors really interact within pancreatic beta cells? How does hyperglycemia and these different inflammatory stimuli really, do they have any crosstalk, or how do they influence each other? So we're looking to see, first and foremost, does high glucose alone actually elicit an inflammatory response from pancreatic beta cells within our model systems? And do pro-inflammatory cytokines in our model system affect these glucose responsive elements, things like TIG-SNP or amylin, in pancreatic beta cells? So we started with our INS model system. This is a rat insulinoma cell line. So this is a cancer tissue generated from beta cells. So they do have a little bit of both phenotypes and functionality. What we see is that when we start treating with low glucose, this 5 millimolar low glucose, it really doesn't matter if we treat them with or without cytokines. There isn't really a change in expression of some of these inflammatory associated genes, things like nuclear factor kappa B, this NF-kappa B, or interleukin 1 beta. Cytokines alone aren't having an effect. And to clarify, the cytokines we're treating with are a mixture of interleukin 1 beta, interferon gamma, and TNF alpha. And alone, they don't really have much of an effect. However, when we repeat this, but in a high glucose concentration, this 20 millimolar glucose, we see that adding cytokines causes a significant increase in the expression of these two genes, suggesting that there's some type of pro-inflammatory phenotype these cells may be taking on. At a protein level, we've followed pretty much the same treatments within our cell lines, and we see that pretty similar expression. We have an increase in NF-kappa B when we treat with both combination of these stressors, and another inflammatory associated protein involved in inflammasome formation, this NLRP3, is also increased. And again, it only is increased when treated with both a metabolic and pro-inflammatory stressor. This suggests to us there's some kind of link here between these two processes, that we require both to really have this potentially detrimental effect on beta cells. We did reproduce this in islets to some extent, just due to the fact we're using islets sourced from IIDP and we're kind of at their mercy as to when we get shipments. We only have an N of 2 right now for our gene expression. However, our preliminary data for gene expression follows the same trend, where when we treat our islets with a combination of both our 3 cytokines and high glucose, we, again, preliminarily have this increased expression in interleukin 1 beta and NF-kappa B, as well at a protein level where we have had enough samples to be able to do some statistical testing here. Same trend applies. We have an increase in NF-kappa B as well as NLRP3 that only seems to occur when we have a combination of these two stressors, suggesting these need to happen concurrently to have an effect in the case of type 2 diabetes. In the case for more extreme hyperglycemia, where we're looking at maybe a 30 or 35 millimolar glucose, other reports have shown that alone is sufficient to induce interleukin 1 beta secretion. But again, we're looking at a slightly lower and generally what's considered a more physiologically relevant glucose concentration. And for anyone in the audience that works with immunology, you're probably wondering, this is just protein and gene expression. A lot of these require some type of processing to really have a functional form. And what we've done here, though, well, we haven't had a lot of luck with phosphospecific antibodies, cleaved and uncleaved forms. We have figured out that, well, nuclear factor kappa B, as the name nuclear factor probably implies, acts as a transcription factor. So while we might not be able to tell if it's phosphorylated or not, we can at least tell, is it in the area of the cell where it's carrying out its function? Is it activated to actually have an effect within these cells? So again, within our INS rodent cell line, we just stained for two different markers. We stained for DAPI nucleus in blue, as well as in red, we stained for our NF-kappa B to see where in the cell is this protein localizing to. Because if it's within the nucleus, that would suggest to us that it's actually having an action there and inducing the expression of other inflammatory associated proteins or genes and then subsequently proteins, and overall contributing to this inflammasome and pro-inflammatory state of these cells. By doing this cross-sectional analysis, we could see that we have this area in blue in our middle of our graph where our nucleus is. So this is everything that's within the nucleus. Well, we also have these cytosolic regions that are still stained for our proteins, but they're not within the nucleus. And specifically, our red staining is for NF-kappa B. So in the case of our low glucose treatments, we could see there's very, very little of it within the nucleus. The majority of our NF-kappa B localizes to the cytosol when we treat with 5 millimolar glucose. And as a nuclear factor, it's not acting as a transcription factor when it's in the cytosol. In contrast, when we treat with both our 20 millimolar high glucose in combination with our cytokine cocktail, this pattern changes a bit. We still have some NF-kappa B that remains cytosolic, but quite a bit more of it is now within the nucleus, where it can be acting as a pro-inflammatory factor and contributing to this inflammasome formation, suggesting to us that while we do have this both gene and protein level increased expression under both of these conditions, there's likely also a functional component as well, where having just an increase of them is also contributing to having an actual effect within this cell. But that's only one part of the picture of the development of type 2 diabetes. Another one that comes into mind is looking at amylin. So with the case of amylin, it only really has this hormone function when it's in its monomer form. This is what it is able to circulate as and contribute to regulating other islet cell function. However, in the case of a more age-related type 2 diabetes, we see that this amylin begins to form oligomers. So in the case of type 2 diabetes, we could see that amylin starts misfolding and aggregating. Granted, this was a study done back in the 90s when the distribution of type 2 diabetes across demographics looked a little different, but upwards of 90% of individuals with type 2 diabetes at that time were found to have amylin plaques within their pancreases, suggesting that this has some role in an age-related type 2 diabetes development. And furthermore, previous work from our lab has shown that in individuals with type 2 diabetes, they have decreased proteasome function. The proteasome is more or less this recycling center of the cell. It's what helps break down any proteins that are misfolded or no longer needed, possibly by the cell, and contributes to creating these more or less subunits to be able to build new proteins for the cell to actually use. In type 2 diabetes, we have a decrease in proteasome function, and furthermore, these amylin oligomers on their own, regardless of diabetes state, are actually able to decrease proteasome function as well, suggesting that somehow this amyloidosis and proteasome dysfunction are likely linked. As these protein aggregates form, it does contribute to cell stress and ultimately cell failure and loss of insulin secretion and possibly cell death. While we know that there's this link between the two, we wanted to expand on this and went on a little bit of a fishing expedition and did some RNA sequencing just to see what effects does proteasome inhibition generally have effect on gene expression within beta cells. Are there any particular pathways that are really affected by this? So we already know in type 2 diabetes, based on previous publications, that we have a decrease in proteasome function. And we are able to mirror this by treating with some proteasome inhibitors, specifically the compounds lactocysteine or bortezomib. We're able to reduce proteasome function and therefore be able to kind of mimic this type 2 diabetes stage within pancreatic islets. All of our sequencing here was done within islets provided by IIDP, so again, we're using human primary tissue. What we found was that when we start clustering these different samples based on just their expression of different proteasome-associated genes, some interesting patterns emerge. So our two middle groups here are our control and high glucose treated islets. So these are ones from non-diabetic donors. And with this, there's not really any huge changes in our proteasome associated genes. There's not really there's maybe some upregulation, largely a downregulation. We don't have much of a change. However, when we treat acutely with our proteasome inhibitors, we have a pretty marked upregulation of a lot of these proteasome-associated genes, suggesting that there's some type of compensatory effect happening. When we have less proteasome function due to a pharmacological agent, the cells start producing more proteasome to compensate for that. What becomes interesting then is looking at our type 2 diabetic individuals. When we have islets from donors with type 2 diabetes, we kind of have this midpoint kind of mesh of these two different genotypes. We have some upregulation of some genes, some downregulation, suggesting to us that maybe acutely, beta cells are able to ramp up production of select proteins to try to compensate for this protein stress. However, in a more chronic state, they lose this ability and actually start to fail in that regard because they're not able to keep up with this different stress. In addition to looking at just proteasome-associated genes, we also looked at quite a few different proteins and just pulled a few here of interest to us that were downregulated in both our acutely proteasome-inhibited samples as well as in type 2 diabetic islets. Some of them are ones that are pretty important for beta cell function, for instance, amylin, as well as the transcription factor Th1nk. Furthermore, we saw a decrease in some of the proteins that are associated with being able to actually carry out a lot of beta cell functions. Things like being able to form insulin granules or regulate kind of the glycolytic state of these cells and altering that and therefore the metabolism, ATP production, and subsequent insulin secretion. They also lose a lot of the transcription factors that really give them their functionality and phenotype. We have a loss of insulin transcription factors, a loss of beta cell transcription factors, and even going far back as neuroendocrine and pancreatic transcription factors. So they're losing a lot of these components of their phenotype that's contributing to, well, really their function and identity. So for our further experiments, we really honed in on these glycolytic associated proteins and looked at glycolytic flux. To do that, we started seeing what steps of this pathway might be affected by proteasome dysfunction. So do we have changes in the glucose uptake, which, given that we have a reduction in our glucose transporter, we would kind of expect, unless there's a compensatory effect of some kind? Do we have changes later in these metabolic pathways, looking at pyruvate production? Or do we even have changes to mitochondrial function? Do we have changes in mitochondrial membrane potential, for example? And how might this be affecting cells and their subsequent ATP production? We began by testing these and kind of building our assays within our insulinoma cell line. So we looked at just glucose uptake on its own, whether we're looking at low glucose, high glucose, or our proteasome inhibitors. And as we could see, when we treat with proteasome inhibitors, we have a reduction in glucose uptake. So this tells us that, well, they have less glucose coming in. That's not surprising, given that we see, at least in human islets, there's a reduction in GLUT2, our glucose transporter. Furthermore, we have a subsequent reduction in pyruvate production. This could be due to the decrease in glucose uptake. However, it could also be suggestive that some of these later steps in this pathway are also impaired. Furthermore, we did some staining with the JC1 mitochondrial membrane potential dye. And what we saw was, again, we have a pretty sharp reduction in the amount of really mitochondrial membrane potential we have when we start treating with these proteasome inhibitors. And this is just a ratio between the aggregate form to the monomer form of this dye. So when we have this green stain, that generally tells us we have a lower mitochondrial membrane potential, a lot less activity happening there, compared to our 20 millimolar glucose sample here with our red staining. Shows us there's a lot more activity and a higher potential. And again, we have a reduction when we treat with our proteasome inhibitors. This could be due to the decrease in glucose uptake. However, it could be due also to multiple steps along this process being impaired by proteasome inhibition. This was done within our INS cell line, so we have begun testing this in human islets as well. And what we've seen so far is the earliest stages of just glucose uptake are reproducible in human islets. When we treat with a proteasome inhibitor, we do have a reduction in glucose uptake. And we have begun also doing mitochondrial membrane potential staining. And while we only have an N of 1 right now and don't have quite enough to do statistical testing, visually it becomes pretty clear we have a lot less of this red staining, suggesting a high mitochondrial membrane potential when we look at our proteasome inhibitor treated samples. And despite everything we seem to know about type 2 diabetes right now, there's still a lot we don't know about the treatments. A lot of the earliest interventions for treatments and kind of the first line for this are looking at lifestyle modifications, things like changes to diet or exercise routines in order to try to manage glycemia. With this doesn't work for everyone. There's issues with compliance, as well as when we think about what functional beta cell mass is actually there. As this disease progresses, there just might not be enough beta cells to really be able to keep up with metabolic needs of an average person. So this is something that doesn't always work for everyone, and that's why we have pharmacological approaches. Some of the older generations, things like metformin, sulfonylureas that most people have probably heard of before, have fairly good efficacy. However, they do carry some risks with them. The sulfonylureas in particular have been associated with some cardiovascular adverse events, and both have been linked with some gastric disturbances. So there are some side effects and risks associated with them. And we do have our newer generations that have made headlines in the last few years, things like the Ozempics of the world, all of these. They do raise, and even though these are great treatments for the time being, they do also carry some issues. There have been issues with accessibility to the medications due to the fact that there have been shortages previously, as some of them have also been prescribed for weight loss. There's some competition in the market in order to actually be able to get access to some of these. And one of the earlier studies looking at these medications back in 2019 actually showed that up to about 25% of the weight loss while on these medications actually comes from skeletal muscle. And if we're thinking of type 2 diabetes in a more middle-aged population, we start to wonder if that's really a good time in life to be losing skeletal muscle. Generally, if we start looking at individuals that are in their 50s or 60s, if you start losing this lean muscle mass, and this lean body mass, you can be contributing to things like sarcopenia that maybe 10, 15 years down the line contribute to the risks of things like falls and injuries, especially within the elderly. So these aren't perfect medications. And even though we have all these treatments, there's an estimation that about half of individuals within the United States and Canada will fail to achieve their glycemic goals. They'll continue to have this hyperglycemia, and contributing to these potential complications of it, things like nephropathies, neuropathies, so on and so forth, that really lower quality of life for a lot of people. Given that there's still room for improvement within these treatments, we have worked with a biomedical engineering lab to see what other mechanisms are out there to treat this disease. This lab works with therapeutic ultrasound. Ultrasound itself is this energy waveform that exists at a frequency above our hearing. We hear down the acoustic range. Ultrasound's a little bit higher. And usually when people think of ultrasound, they think of the imaging modality, things like an ultrasound for a pregnancy or an echocardiogram. Therapeutic ultrasound's a little different. Instead of trying to image something, it's actually trying to elicit a response from its targeted tissue. Specifically, by adding these energy waves effectively, we can create pressure and deformation of a plasma membrane in order to elicit a response from the tissue we're targeting. And with this, we've found that there's kind of this ideal range where you could treat pancreatic beta cells and actually get an increase in insulin secretion from them. So specifically, we can get some pretty large increases in the insulin secretion from these cells. This was done within a Th1nk cell line, so a similar rodent insulinoma cell line. However, even though we have these really great increases in insulin secretion, we actually have to work at a higher frequency because these two that are kind of the ideal for insulin secretion actually cause a lot of tissue and cell damage. So we moved to working down in our 800 kilohertz range. And with this, in the Th1nk cell line, it's not significant in increasing insulin secretion. However, this has been replicated in vivo in mice, in vivo in rats, in ex vivo rabbit pancreas, as well as in human pancreatic islets, we do have a significant increase in insulin secretion. So the engineers kind of went through and fine-tuned what range are we really working at, and what's the system to prevent heating, prevent tissue damage for these cells. My job then was to figure out what's it actually doing to these cells and how is it doing it? Why would an internal organ like a pancreas, really in general, need to be able to detect therapeutic ultrasound or any type of pressure? And while we know insulin secretion occurs, what other effects does this therapeutic ultrasound have on cells? And can these additional effects be replicated in vivo? Well, given that we're looking at a calcium-mediated exocytosis, we wonder, is this ultrasound somehow creating a calcium influx into these cells? And that's where this idea of mechanosensitive channels comes in. These are plasma membrane channels that actually function as cation channels when activated and allow for this calcium influx that can then influence insulin secretion, kind of independent of glucose. Going through the literature, we were able to find that in different model systems and different tissue types, whether it be exo or endocrine tissues from pancreases, there's quite a few different mechanosensitive channels expressed. Within our INS cell line model, as well as our human pancreatic islets, we were able to find expression of 3 different forms: this transient receptor potential subtype phenolloid, 2 and 5, as well as Piezo1, are all expressed in both of our model systems here. And within our INS cell line, we then assessed what other changes are occurring when we treat with therapeutic ultrasound, or TUS abbreviated here. What we see is that both amylin and insulin are significantly downregulated when we treat with therapeutic ultrasound, which raises a bit of a red flag because this is something that would suggest maybe the cells are being harmed. And we want to make sure this isn't what's happening. So we also want to figure out, how is this mechanism happening? So we tried treating with a few different compounds. These great names of Yoda and Doku are actually specific agonists and antagonists, respectively, for the Piezo1 channel. And ruthenium red is this broad inhibitor of all mechanosensitive channels, so our TRPV channels as well as our piezo channels. When we treat with both ultrasound and this ruthenium red, this broad mechanosensitive channel inhibitor, we actually are able to really prevent this downregulation of amylin and insulin, suggesting that this downregulation is occurring through these mechanosensitive channels. Finally, we tried to replicate this in vivo and got some slightly different and interesting findings. I should specify this was done in a Zucker diabetic fatty rat model. And we then took just some histology slides, really, and stained for insulin, amylin, PDX, and tried to see what changes we were able to find. And when we treat with ultrasound, we actually don't have changes to insulin and amylin within these islets in a live animal, which is very different from the findings we see in our cell lines. What we believe is happening, and we're still trying to work out the details of, is that there may be some degree of macrophages actually coming in and remodeling this tissue in order to actually compensate for cells that may be damaged throughout this process. Even if the viability looks good, it doesn't mean this viability we're getting measured through things like trypan blue might show the cells are okay, but they still might have functional damage that is not being assessed through that. So with that, we're able to see that there's a change there, and we do think it may be due to a tissue remodeling factor. Interestingly, we do have a decrease in PDX1, a pretty essential transcription factor for these pancreatic baked cells. We could see in our sham-treated lean animals, we have this nice border where we expect beta cells to be that have a really strong staining for PDX1 that's immediately lost when we treat with ultrasound or in animals that are obese and confirmed to have a type 2 diabetic phenotype. So from there, we're able to see that maybe this reduction in insulin and amylin that we see in the INS cells might actually be due to a reduction of another transcription factor like PDX1. And furthermore, as this protein is actually lost in these cells, and maybe this is an indication these cells are stressed. And as that occurs, though, there's likely some type of immunological remodeling happening that allows for these cells to maintain functionality throughout the whole pancreas. I know there's a lot there, and I'm happy to take questions, but with that, I just want to quickly acknowledge everyone who's worked on this project, because given how much has gone into it. There's a lot of people to thank, whether it be our current lab members, our previous lab members, especially Dr. Andrew Chen and Dr. Didi Chaudhary-Bhowmick, whose works I've largely built on, and to thank the PIs for this project. From the biomedical engineering side, Dr. Vesna Sidarich, as well as my primary PI, Dr. Alexander Yaramich. And with that, I'm happy to take some questions. I could pull up the chat here and see if there's anything.
Yeah, great talk, Mallory. We do have a question from Julia Wang. Very cool talk. I'm wondering, how do you do ultrasound in animals and directly target pancreas?
Yes, so for the animals, these are anesthetized rats. So we actually initially were using it's an inhaled anesthetic. We ran into some issues with that, where apparently that can cause changes in blood glucose on its own. So we moved to using propofol to anesthetize rats. And from there, we did some imaging to get an idea of where the pancreas is within these animals. So doing a little bit of an imaging modality as well. And with that, more or less just going through and quite literally with a Sharpie, tracing an outline on a shaved stomach to see this is the area we believe we're targeting, and this is what we're going to try to. From there, we're just then switching transducers, moving to a therapeutic ultrasound and treating that area. It would be ideal if we could use both imaging and therapeutic at the same time, but given that they're both in roughly the same frequencies, we run into some problems with them interacting with each other. So it's kind of a little bit of both. We have to do step 1 of imaging, step 2 of treating. But that is a good question.
As a follow-up question, do you need to adjust the ultrasound levels in vivo and in vitro?
Actually, we only slightly, so we're still using the same frequency and same duration. The only difference is when we're working with a nice little 96 well plate, we can place it in a water bath while we're treating to prevent any thermal effects. When we're treating an alive animal, we can't exactly do that. What we usually try to do is pretty much just cool our ultrasound gel so that it's cold enough to start with that we can reduce thermal effects where we could potentially burn something, and that's not what we want to do. And during the entire process, we just have a temperature probe there to make sure it's not heating too much. Occasionally, usually if we're treating multiple animals in a day, because I think we had 12, we were treating 12 a day. So usually by like animal 8, 9, 10, even though we'd have this ultrasound gel cooled, we still start seeing a thermal effect. We'd switch over to pulsing the ultrasound and having it on for half a second, off for half a second, on and off, which ideally we wouldn't have to do. We tried to rotate animals to minimize the effect of that. So which order we were treating the animals in, we tried to rotate, but we do run into some issues with that due to thermal effects.
Great. Also, Anupar Nagiri would like to ask a question.
Yeah, well, nice talk. So my name is Anupar Nagiri. I'm in biological sciences at East Tennessee State. I've been studying proteostasis and protein folding for about like 15 years now. So I'm obviously interested in your proteasome inhibition, that part. So I have a bunch of questions. To start with some technical questions, then I'll go to the science questions. So
Sure.
when you said you use the insulinoma cells and you specified, you wrote in cells, but can you tell us which INS one? Is it INS 1 or inside 30 to 13?
Yes. Oh gosh, I've just left the lab like a month ago, and I'm trying to make sure I'm telling you the right order of things. It's an INS1 derivative. Is it INS 13/832?
830 to 13? Okay.
Yeah, it is based off the INS1 cell line though. It's just a minor derivative of it.
Okay. Great. And then another technical was about the mitochondrial membrane, I think. Yeah. So in this one, the third graph, which says aggregates upon monomers of what? Like which protein?
So
are
it's
we talking
actually, it's actually not actually of the protein, but of the dye itself. This dye actually works ratiometrically
about?
so that we have this green versus this red, and we're able to kind of see, do we have pretty much when the mitochondrial has a higher potential, this dye will kind of trap itself within the mitochondria so that in this high concentration, it'll start to form these aggregates of this dye. And that's what gives it this red color. While when it's just kind of diffused out in the cell, it's less concentrated, therefore it can stay as more of a monomer and keep this green color. So
Okay.
it's not necessarily of the proteins, but of a ratiometric dye.
Do you know the name of the dye?
JC1.
JC1? Okay,
Yep.
thanks.
We've had a lot of, I will say, we did have, while it worked beautifully in a nice cell monolayer, we do run into some issues loading it into islets. Generally, we do have to do quite a bit longer incubation times, and the next graduate student coming in is suggesting using some EGTA to kind of partially dissociate them a little bit to allow better penetration into the core of the islets. That is my disclaimer to them.
Yeah, but that's good to know. Now coming to the science questions. So amylin aggregation, IAPP aggregation, you showed in one of your cartoons pictures which indicated impairment of proteasome function.
Yes.
That one. So do you know how?
We're still trying to actually establish that. What we know right now is that the monomers, they can be degraded by the proteasome, but don't affect its functionality. And the aggregates actually, like full-blown aggregates and plaques and fibrils, don't actually interact with the proteasome at all. Only the oligomers seem to. We know it interacts. We know it is within I should specify, from a colocalization microscopy, we know it interacts with one of the beta subunits of the proteasome. I did some immunoprecipitation as well, where we're not seeing an interaction with the alpha subunits, though. So it seems to be specifically interacting with the beta subunit, but the specifics with that is still a little to be determined. And we know it does have an effect on amylin itself, has an effect. Well, actually, not amylin itself. The amylin oligomers have an effect on the trypsin-like, caspase-like, and chymotrypsin-like functionality of the proteasome.
Okay, got it.
Yeah.
And do you know where IAPP aggregates inside the cell? Is it in the endoplasmic reticulum, cytosol?
That honestly, there seems to be, we don't know at which stage it begins forming oligomers, but we do know that these oligomers and aggregates are found throughout a lot of the cell. We see it, like you said, in cytosol, endoplasmic reticulum, and we even see it within the nucleus, which is a little odd too. Yeah, we see it pretty much throughout the entire cell. When we're looking at full plaques where we have oligomers upon oligomers, those are actually mostly extracellular, largely.
Oh, okay.
Yeah, for some, we do see a few intracellular, but those are largely more extracellular. Those are things we'll see more in like in a pancreatic section, more histology type staining.
Did you happen to look at these aggregates or anything at all with respect to the subcellular localization of IAPP in your type 2 diabetes IIDP?
Unfortunately, no. We
No?
really were only able to work with the RNA we had from these type 2 diabetic islets, just because we get about one shipment of them a year at best. So we have to work with whatever we have stored. And unfortunately, these proteasome assays do require a good number of cells, and doing something like subcellular localization of it's going to require a lot of cells as well. So we're a little resource limited in that.
I see. Last question. I know I don't want to take up.
No, of course.
Yeah. So you found from the RNA-seq results, there are some genes that you showed that were downregulated under proteasome inhibition.
Yeah. Oh, this is just a few key ones that we've picked out for future analysis, but there's quite a few.
Do you know, or if has anyone done this wherein they have IAPP aggregation leading to proteasome inhibition and then an RNA-seq?
So basically just like treating the cells with pre-aggregated amylin, more or less.
Or inducing them to make them oligomerize.
Okay, yeah.
And then see if proteasome is inhibited.
I haven't seen RNA sequencing data for that. We do know that we also have the data is not shown here. It's for a little bit of a different project I was helping an undergraduate with, but we do have a INS cell line that is transgenic for human amylin. So it overexpresses human amylin. And in that cell line, we do have a decrease in proteasome function across all three enzyme-like activities. So we do know that there's something there. An RNA sequencing experiment has not been done to the best of my knowledge.
I see. Okay,
Yes.
thanks for patiently answering all my questions. It's great talk.
Of course. Thank you.
That was really great discussion. I just wanted to back up just a little bit.
Sure.
Therapeutic ultrasound is normally used in patients, right? For soft tissue healing, like they have pain relief, bone healing.
There's actually, in addition to that, there's a few other, it's almost I don't want to say a destructive type of therapeutic ultrasound, but it can be used to break up kidney stones as well as for different types of ablations. The big
So
one
it's
that they've
a
been,
matter of the frequency, right, across the
yep,
scale?
frequency and intensity. Yep.
Mm-hmm. Okay,
Yeah.
yeah, so that's so interesting. So you kind of really tuned in on this, very, I mean, I guess therapeutic ultrasound, but a therapeutic bandwidth to kind of
Hmm.
interface with the channels and the inflammasome. And do you think if you could get, even, more specific on your therapeutic wavelength and, whatever,
Hmm.
whatever it is you're trying to tune in on,
Yeah.
could you get even different results depending on how you're, expanding or
Yeah.
contracting that?
Yeah. We honestly, I will say, I probably should have linked more of the papers here. This biomedical engineering group, this Darich lab, has been working on this for quite a while. I think they started this grant back in 2015. They've
Okay,
done a lot of the heavy lifting for figuring out what can we treat with, what frequencies, what intensity, what durations,
okay,
and really narrowing that down just because when we start treating at something, given we see 400 kilohertz, there's more insulin secretion.
right,
Who's to say 100 kilohertz doesn't have even more? The issue we run into is when we start staining with trypan blue, we have huge losses in cell viability. I believe the 400 kilohertz was about a 60% decrease in cell viability. So
yeah.
we have to kind of balance this.
You need
Do
the
we want more insulin
Goldilocks.
secretion or do we
Yeah,
want, yeah. So we actually are able to work in a pretty narrow range. There's another student who's currently working at a 900 kilohertz. They're working on a slightly different system in a different tissue that has more thermal inputs where having that heating could be a problem. So they've scooted down or scooted up in frequency and down in intensity. So we're able to actually modulate it pretty well. I'm not sure how much more we could really hone in on that. I think the bigger issue, and this was already asked in one of the questions, is how do we make sure we're targeting the right area?
hmm,
And that's probably going to be the biggest hurdle, I think, for this.
yeah. Well, it's quite interesting. It's novel, and we really hadn't seen it come across our think tank, Ask the Expert
I
collection.
I will say the thing that jumps out to me for the context of type 1 diabetes, that's generally what I always try to come back to
That
somehow.
was my next question.
Yeah, the thing that comes to mind for me is having these mechanosensitive channels means, well, obviously we know the pancreas is a mechanosensitive organ. There's a reason that pancreatitis after abdominal surgeries is a fairly common complication in the grand scheme of things. This is a very kind of more or less touchy organ, even though it's not something that is generally thought to be pressure sensitive. It does raise the question to me as to can we modulate the activity of any of these channels to better improve things like encapsulation of islets? Do we change something like scaffold stiffness or the area between them? I do kind of wonder that a little bit if that plays any role in possibly the viability of these cells. And as we've seen, the activation of them can change things. So does it play a role in maybe their ability to secrete insulin? Is something that immediately comes to mind for me.
They're surrounded by, I mean, so interesting that you brought that up because like Sarah Richardson's really great new work where she detected the loss of these sort of endocrine organs, the smaller
Mhm.
ones early on, right? And they don't have sort of the robust extracellular matrix around them, and they're the ones to sort of be to go first. So that's a great point you brought up, Mallory, and it's just food for thought. For everyone
Mhm.
listening, reach out directly to Dr. Brayer if you would like to learn more. And she is right now on the job market as well. So, and she can be reached at
Yep, feel free to reach out.
on
I'm also on
LinkedIn.
LinkedIn.
All right,
Wherever
sounds
people can
great.
find me.
Thank you, Mallory. Really appreciate this great talk and have a great rest of your day.
Thank you so much. I appreciate the opportunity. Thank you.
Thanks. Bye-bye.